Pending. Wow. What does that mean?
We're going to work out all the kinks today.
There we go. Excellent. Well, good morning and good afternoon, everybody. Welcome to the first of our Grants4Ag seminars that we're hoping to host over the next few months as part of our Grants4Ag program. For those who don't know me, I'm Phil Taylor with Bayer Crop Science based in St. Louis in the US, and it's my privilege and pleasure to sort of moderate the session today. Just like to start very quickly by telling you a little bit about our Grants4Ag program. This is our early innovator seed funding where we provide a small amount of funding to academics and startups around the world to help them take that first step in understanding if an innovation or an idea that they have has application for relevance for agriculture, for our business and our R&D pipelines. And so last year we were excited to fund 24 projects through this program, and we're hoping over the next few months to showcase some of those for you here today. So just captured on the slide, you can look at it at your leisure, some of the features of our program, where it's designed to be straightforward and low touch and not burdensome at all, and hopefully allows you to do the great science that you're doing and maximize the opportunity. But as we talk to the awardees and our new sort of cohort of friends that we've developed through this program, we talked a lot about ways we could help them showcase their science and ways we could sort of give back at large. And so we created this seminar series, and I hope you enjoy what we're doing today. And obviously any feedback and thoughts are very welcome. But it's my great pleasure really to kick this off and introduce Maria de Rosa. So Maria has been a wonderful part of our program this year and always volunteers to do the first something. So I super appreciate that. But Maria's going to spend some time talking to us today about the work that's going on in her lab at the Department of Chemistry at Carleton University in Ottawa, Canada, where she works on aptamers, a really novel type of molecular tool. And what I'm excited about to hear from her research is how she's thinking about how those can be applied both in agriculture, but in medicine and nanotechnology and all kinds of different fields. So super, super cool science and super cool research. A little bit about Maria. She received her bachelor's and PhD both from Carleton University and there in Ottawa, Canada. So I guess she really likes being there, I suppose, and has returned there as a faculty member in 2005 and has been there since then, and is now the interim dean for the faculty in science. So really appreciate her taking her time today, because I know she must be very, very busy with all that kind of stuff. So Maria, the floor, I will stop sharing, and if you would like to share your slides, and the floor is yours.
Thank you so much, Phil, and everyone at Bayer. It's really a delight. Thank you so much for allowing me to share my research with everyone today, and for supporting us here at Carleton through the Grants4Ag program. Okay, let me start by sharing my screen, and we always start the same way by asking, can you see that? Just let me know if once you're able to get a view of my slides. Perfect. I think we'll probably keep doing that even when we're back in person. Can you see my slides? We're all in the same room. So, thank you so much for the opportunity to talk to everyone today about the work that we're doing, as you mentioned, at Carleton University. We're up in Ottawa, Canada, and the work that we're doing, as you heard, is related to a really interesting, exciting molecular tool called aptamers. And I'm going to focus on application of aptamers in the space of agriculture and food, but you're 100% right. This technology, we're using it for things like delivery of medicines, targeted delivery. We're using it for diagnostics. We're using it for all kinds of different things. But we were really, really excited when, we saw on Halo, however many months ago, this opportunity to kind of think about agriculture in a different way, because we had already started imagining ways that aptamers could influence this really, really big field and solve some of the problems that are inherent with agriculture as we move forward. Okay, so let's get started. I just want to give you a little bit of background in the field that I'm working in. The idea of using DNA in this unusual way. We're not talking about the biology of DNA. We're not talking about genomics, but DNA nanotechnology. And the idea is that DNA nanotechnology uses DNA for its kind of privileged structure, this really amazing backbone, bases, molecular interactions built in to this really cool molecule. And so I'm showing you a quote from a chemist, a Nobel laureate, Roald Hoffmann, who in 1994 wrote this really great piece called DNA as Clay. And this has been inspiration for the field, since that day. And you can see the quote, I don't need to read it, but I'll just sort of summarize the idea that DNA has been optimized, to be this really interesting molecule that's able to do some really amazing chemistry. So let's use it to do something new, something beautiful, something useful, right? And that's what we're hoping to do in the space of agriculture. Okay, so a little bit more about aptamers. You're looking at this little squiggle here on your screen. This red ribbon is representative of one of these aptamers folded up to bind to a target. That's that blue molecule. So what are they? They're basically synthetic DNA or RNA typically, and they fold up into these nanoscale objects basically. And they have built in this inherent ability to bind to targets that are not just nucleic acids. So we're all sort of aware that DNA will bind its complementary sequence, but this is a little bit more than that. The DNA folds up into a pocket, basically. And you can see that here. This is called a hairpin structure. And up here, there's a loop that's folded over. And there's the ability for this molecule to interact with a different molecule through all sorts of non-covalent interactions. So we're interested in understanding those interactions and exploiting them for all kinds of different affinity-based technologies. So a little bit more about aptamers. You can think of them as this lock and key in many cases. In some cases, the key is the molecule, so the aptamer forms that lock, and only one key can fit in. And in other cases, say for a bigger target like a protein or a virus, it might be that the aptamer is the key. It fits into a specific cleft very specifically and with high affinity. So binding with high affinity is one of the key aspects of this technology that makes it useful. Low levels of target are able to be potentially detected by these nucleic acids, which is so important. And also the level of specificity that's possible. So the idea that if we want to use this technology for any kind of recognition, then there needs to be that level of specificity or selectivity. So this aptamer will bind to target X and not target X with a very small change associated with it. And the example that typically gets used when we talk about the specificity of aptamers relates way back to one of the first aptamers that was generated for a molecule called theophylline. And theophylline is basically a drug, but one small change in the molecular structure of theophylline makes caffeine, which is one of our favorite molecules. But in a way, it's so interesting because the aptamer that binds theophylline is 10,000-fold less able to recognize caffeine, which is just one small molecular change, that hydrogen turns to a methyl group, shuts off binding. So that kind of level of specificity, you can imagine there could be a lot of interesting applications for that. So up here in Ottawa, Canada, what are we doing that relates to aptamers that maybe you'd be interested in hearing about today? We have basically a pipeline where we're discovering new aptamers, characterizing those affinity interactions that I mentioned, and then applying them to all sorts of different problems. So, in terms of the aptamer discovery, we're really interested in the agriculture and food space. So we're interested in finding aptamers that could be useful for things like food safety diagnostics, delivery of nutrients to crops, which we'll talk about today, etc. In terms of characterization, we are a chemistry lab, so we're really interested in understanding at the fundamental level what's going on with these aptamers and how they're interacting with their targets. And then applications, like I mentioned, they range from things like simple dipstick tests that could be used out in the field all the way to MRI contrast agents and drug delivery vehicles. Along the way, we have some fun. So this is a little gift that's a snippet from my group's Dance Your PhD entry many, many years ago now that won Science Magazine's contest, where they danced the process that's called CELEX that we use to find new aptamers. So I'm going to explain that a little bit in a second once I get through a little bit more intro, but aptamers are not designed, they are discovered. And so if you're interested in how that process works, I'll explain it in chemical terms in a minute, but I showed my mom the dance and she says she understands it now. Okay, let's take a step back and now think about the big picture that I want, the picture I want to paint for all of you guys. The idea that aptamers could play a role in agriculture writ large. So we know that there are big challenges facing agriculture moving forward. We have to feed, so many people on shrinking land with climate change, smaller resources, smaller footprint. How can we, really tackle these challenges? Well, there's a movement in agriculture that's focused on, using data, right? Informing decisions about how to manage crops and using that to improve efficiency and quality. And so this is precision agriculture. And this is just, a cartoon that I'm showing the idea that, well, we've got a crop and there are sensors and they're interacting with drones and the drones are going back to the hub. And there's all kinds of, information that's helping farmers make decisions about how to treat their fields in order to maximize their efficiency. But currently, what does that sort of look like? It's abiotic for the most part factors that are really coming into play with precision agriculture. So looking at salinity, looking at pH, looking at temperature. There's definitely, can we also see things like pests and that sort of thing. But at the molecular level, maybe there's an opportunity for us. Let's imagine that we could include this idea of molecular information in our toolkit for trying to make decisions about crops. So that's what I'm going to talk about today, the idea that aptamers can be used to decode some of the signals, the inherent signals that are present in crops, in the rhizosphere, for example, and using that information to respond. For example, respond with nutrients, respond with treatments, these kinds of ideas. So what you're looking at here is a little cartoon, and I'll explain more in detail, but just the idea that the crops are releasing signals into the rhizosphere. We can, and those signals are related to all sorts of things: nutrient status, the need for protection from herbicides from other pests, all sorts of things. So if we can use a molecular affinity tool to detect those molecules and then use that information to inform decisions about how to treat the plants, we could be able to really add more information to what is already available through traditional precision agriculture approaches. So that's one part of what we're doing. And then I hope, if I have time, to talk to you a little bit about some of these other things. So the idea that aptamers might play a role in delivering some of these treatments to the crops. Aptamers can play a role in just even the basic understanding of things like, toxin levels or other factors. And that aptamers might play a role in responding to the environment. So stress inhibitors, that kind of. All right. So big picture, we want to use aptamers for all sorts of things related to agriculture. First off, where do these aptamers come from? And I mentioned there's a process called SILEX, systematic evolution of ligands by exponential enrichment. You don't need to memorize that. But the idea is that there's a screening process that allows us to look through and kind of filter through billions and billions of nucleic acid sequences just to find the ones that do the job we're interested in. So we start off with an initial library of sequences that we synthesize, and this library is very big. So 10 to the 14, 10 to the 15 random sequences. And in some way, we incubate those sequences with whatever the target of interest is. In this case, it's a mycotoxin I'm showing you, but it could be anything. And after we do that incubation, we have to find some way to partition binders, sequences that do interact with the target, from non-binders, ones that don't. Majority of the library is not interested in binding to our target by any means. So we have to use very, this is a key step in this process. The stringency of the step really dictates the success of the aptamer selection. Then, we do some amplification, we regenerate our enriched library, and we repeat the process over and over and over again until we've narrowed down our candidates to just a few top sequences. Then we characterize them. But let's just take a quick look at this process. Why is this? Why are aptamers so versatile and so exciting for all sorts of different technologies? Part of it is the level of control that we have over the selection process, which we may not have in other molecular recognition type entities like antibodies, for example, which are generated in vivo. We do all this on the bench. So how is that an advantage? First off, the library that we use to start off the selection, we can design it, we synthesize it ourselves, we can do chemical modifications. So we have a lot of control over what's going into our selection process. And if the aptamer we want isn't in that library to start it with, we're never going to find it. So having a control over that step is really important. We can control the binding conditions. As I mentioned, we do this on the bench. So if someone needs an aptamer that binds at a certain temperature, certain salinity, certain soil conditions, who knows, we can match that selection event to the application conditions to basically improve our chances of finding a good hit. And then that partitioning step that I mentioned is so important because we can tune selectivity at this step. If we know we don't want to bind to molecule Y, but we really need it to bind to molecule X, we can do counter selections to remove binders for those competing species. If we know we need a certain level of affinity, we can control the stringency of our selection so that we can hit that range of affinity that we need. So there, again, there's a lot of possibilities there for control. At some point, we do this process over and over again, and we decide, you know what, we have the level of enrichment that we want. So we do our sequencing. Now it's basically next generation or high throughput sequencing that's done. We get millions of sequences, do some bioinformatics, and out pops a few hits. Here again, there are some advantages that aptamers have over other maybe, more biological molecular recognition entities like antibodies. One thing is that the aptamer is a chemical, we chemically synthesize it. So it's straightforward to do modifications in a homogeneous way that we can, mass spec, we can have low batch to batch variability. So being a chemical species makes it a lot easier to work with and to modify. The other thing that's interesting is that this aptamer at its core is a sequence of nucleic acid and has the ability to bind to its complement no matter what. So that brings us some opportunities in the way we design sensors. There could be structure switching sensors, like let go of the complement and change shape to bind to the target. There's also some ideas that this could be used in therapy as an antidote. The complement will outcompete potentially the aptamer, and you can use that when you're trying to titrate dose and these kinds of things. So lots of really, really cool applications can be imagined based on the fact that the aptamer is made of DNA and it's synthetic. Okay, let's go back now to my big picture. I just want to tell you one quick, a little bit about our work that's involved in field tests type things that are using aptamers, and then I'll get into the rhizosphere biomarker aptamers. So we've spent a long time on this, that we've been working on developing aptamers for different mycotoxins, which are these secondary metabolites that come from fungi and can basically be a problem in all sorts of different grains. For example, you're seeing here wheat and corn. So we have aptamers that are able to really at low levels and with high specificity detect these toxins. And we've been building them into really simple field tests, basically strip tests. They look like little strips you might use to test your pool or small mini pregnancy tests, where you're able to just see with a quick yes, no, quick visual test whether or not these toxins are present. And we've had a lot of success actually recently where we've been using our strip tests in blinded samples to detect low levels of these toxins at the regulatory limits. So for example, one toxin in particular, ochratoxin A, can be a problem in all sorts of grains. In certain jurisdictions, the regulatory limits are as low as 0.5 parts per billion. So we were interested, can we use our really quick tests to be able to detect at that level? And so what you're seeing are just a couple of batches of our tests where we have control tests, and there's a dot that fades when the test is exposed to the toxin. And these were in real samples as low as 1 part per million, 0.5 part per billion, in triplicate, that we were able to detect low levels of these toxins using this really, really cheap, simple technology. So we're really excited about the future of doing that for all sorts of different things related to food safety and testing in the field. But the main story I wanted to tell you today was this idea of, can aptamers be used in a way to basically decode signals that could be coming from crops and then respond to those signals? So this is the idea of things like a smart fertilizer, where aptamers are basically looking for certain key biomarkers in the rhizosphere that are linked to things like nutrient demand. So let me show you a little cartoon here that's going to help explain what the concept is. So these blue circles that you're seeing coming down here, I'm going to get that going again. There we go. Those blue circles that are coming down, this is supposed to represent root exudates. And there are, these are chemical compounds that are released from the roots of crops. There are all sorts of different chemical classes. And I'm going to tell you a story about Agriculture Canada collaboration with us, where they decoded some of these root exudate signals and found certain signals that mapped to nutrient needs. And I'll explain that. Now, if we have those signals, can we imagine using them to help with nutrient delivery? So let's imagine this is a coated fertilizer. The orange circles are the fertilizer, and the green, circle around is a coating. If we include aptamers into that coating, can it protect the fertilizer from premature release? And then when those signals that are coming from the roots, those root exudates, are detected that are linked to nutrient demand, only then can permeability change or a controlled decomposition happen that would allow the fertilizer to be released at that point. So that was the question we asked, and this was, now more than 10 years ago that we started this collaboration with Carlos Monreal at Agriculture Canada to ask that question. Here's a picture of Carlos. He's recently retired, but he's still very active in this area. So what Carlos had done was used a series of mass spectrometry techniques and labeling experiments, along with greenhouse experiments for wheat and canola, to try to find basically chemical signals that could be mapped to nutrient demand. So the idea is watching, as the growing season progresses in soils that did not have any nitrogen and soils that did, watching how different chemicals, their patterns emerged over the growing season. I'm going to show you a couple examples. So this is on the left here is a wheat profile, and on the right is a canola profile. The chemical compounds are not listed because this is Carlos's intellectual property, but we are able to, I'll be able to disclose one for you that we've been working on. But the idea is that over the days of the growing season, there are certain signals that have, peaks and valleys in terms of their concentration in the rhizosphere. So that soil, that zone right near the roots. And in some cases, those patterns are very complex, like wheat with several, spikes, and others it's less complicated, like in canola. And what's interesting is that in some cases, not many, the spikes map onto when nitrogen uptake would or could happen. So this is most likely not causation, it's most likely correlation, but it's reproducible. So the idea is that if we track some of these signals, we might have insight into when the crop is ready to take up nutrients. If we know when the crop is ready to take up nutrients, then maybe we could basically use less nutrients, right? Protect the nutrient, only allowed to be released right when it's about to be, when the plant is primed to take it up. And in that way, you could improve nitrogen use efficiency. You can overcome some of the issues that with, for example, when nitrogen is released too soon and it's leached and it ends up in waterways, there's all sorts of environmental impact. And there's also cost to the farmer. So that's the idea. So Carlos was able to detect some of these signals or track some of these signals, and then he came to us in our group to find aptamers that would be able to detect them. So this is, you can learn more about some of this in a recent publication in Journal of Agriculture and Food Chemistry, but Emily Mastronardi was the graduate student who really led this whole endeavor. And the challenges that Carlos presented to us were as follows. We need an aptamer that's going to have moderate affinity for the root exudate that he just discovered. And it has to be moderate because these root exudates are chemicals that are present in the soil, right? Growing season after growing season, microbial metabolism, there are these chemicals that could be present at very low levels in the soil. We wanted an aptamer that would only recognize that signal when there's a spike, because that's what we wanted to be watching for. When you watch for the spike, okay, now the plant is ready to perhaps take up some nutrients. So we designed a selection experiment that really tuned the stringency to avoid binding at those really low levels. But, there are some challenges with that, and these are always listed here. Weak binding affinity could limit our applications. Weak binding affinity means that, it wouldn't necessarily be the sensor with the lowest limit of detection, for example, but that wasn't what we were needing in this case. The next challenge was that the binding had to happen in soil solution, right? This is happening in the soil. So we met that challenge by doing our selection experiments in soil solution extract. So from the farm, we took the soil and we basically used the solution, with washes as the background for our selections. So this was to hopefully kind of mitigate any kinds of competing species or interferences that could impact our selection. Now, what's the challenge with that? Soil is variable, right? If I take the soil from one zone, it might be different than another zone. So that's something that, we have to keep in mind. This aptamer works in one soil solution. Does it work in all of them? And then the last requirement was that there had to be obviously selectivity for the signal compound. We didn't want this aptamer to bind to other similar molecules and perhaps, lead to premature release, for example, of the nutrients. We didn't want that. So we did do counter selections against other similar compounds. But again, the challenge is, what about unknown unknowns? We did what we could, but certainly there may be in certain scenarios counter species that we weren't aware of. So, weighing all the requirements and trying our best to meet the challenges, we did a CLEX experiment to find a series of binders for this root exudate. So the root exudate I can report in this case is L-serine. So in this one case, it's an amino acid, very simple molecule, but interestingly has been linked in other studies to nutrient needs in different crop species. In this case, it was wheat and canola. So we were able to do our selections and find a series of 3 aptamers that were able to bind the target with good affinity and good specificity. So these are just basically isotherms by a technique called microscale thermophoresis. It's not that critical that we know all the ins and outs of that technique, but basically we were able to find micromolar level binding, which is a good moderate binding for this L-serine in these scenarios in the soil solution. But one thing that was important to note is that the aptamer itself, which is, obviously it's a biological, biocompatible molecule, we did have to be concerned about things like nucleases that could be present in soil and could destroy the aptamer before it has a chance to do its job. So to combat that, what we did was we made something called a spiegelmer. And spiegelmer is basically a mirror image of DNA. So instead of using D nucleosides, it uses L nucleosides. So it's basically the mirror image of what a natural DNA would be. Interestingly, when we made the spiegelmer, it was still able to bind to the target, but it was more nuclease resistant because the nucleases did not recognize this kind of unnatural form of DNA. It was still able over longer periods of time to be biodegradable, but not in that short time scale. So we were able to increase the stability of the aptamers from seconds when we were just using standard DNA. These latter experiments are just showing that the aptamer is getting chewed up, to hours and days when we were using the spiegelmer without compromising affinity. Actually, the affinity got a little bit better. So we have these aptamers and spiegelmers now that bind this L-serine, this target molecule that could be a signal for nutrient demand. So what are we doing with those aptamers? Well, what we're doing is we're making these simple tests, like I mentioned, for the mycotoxin case, that could allow, at a very, very basic level, you could imagine taking soil samples and looking for exudates and then responding. It could be very simple. But then alternatively, a more high-tech, perhaps, use of these aptamers, we're incorporating them into coatings for fertilizers, for example, that would allow the coating itself to be smart. It's recognizing when these exudates are at high enough concentration in the environment around the coating, and then responding to that with releasing what the payload is inside. So to talk about that last part, we did a lot of sort of basic science before we got to the point of actually trying to make smart fertilizers. We did a lot of basic science to try to understand how aptamers might be able to change the properties of a coating after they bind a target. So bind the target, does a coating become more permeable? Does it break down? This kind of thing, so that we could use those as a mechanism for delivery in the rhizosphere. So I'll show you a couple. All the references are, they're quite old now. You could definitely go and check them out. One set of experiments, we made these microcapsules where the aptamers were in the wall of the microcapsule. And then we added target to see how diffusion across that wall improved or, was blocked, everything like this. And what we were able to find is that aptamer target binding can make a coating about 10 times more permeable in this case. So that could be a dramatic kind of slow release triggered by the binding of a target. Another set of experiments, we did burst release type capsules. So in this case, so up here, the green is coming from a tag that's on the aptamer, and you can see it's in the wall of this capsule. But in this other set of experiments, the aptamer was still tagged, but now it was inside the capsule as a scaffold. So now you don't see like a thick wall of green, you see kind of this web of green on the inside of the capsule. And in this case, what we found is that if the target could enter that capsule, binding to the aptamer led to the burst of those capsules. So what would happen is that the aptamer couldn't be a scaffold anymore for the walls of this microcapsule. Instead, would kind of collapse on itself to bind the target, and basically you end up with polymer debris, or you end up with actually burst capsules, as you can track. So we knew that there was a mechanism, we're still trying to understand at the molecular level what's going on, but there was potentially a way to adapt the binding of the aptamer to the exudate, to the release of a nutrient. And so we've moved on from there, and we've been able to put together a prototype of a smart fertilizer so this is basically a urea pellet that's been coated with a nanoscale coating that includes the aptamer. Again, the aptamer in this example is tagged with a dye so we can see its distribution across the surface, and it's relatively well distributed around the surface. We've done some greenhouse studies where we're able to see that the aptamer coating is able to protect the fertilizer from release. In this case, it's conventional urea pellet from release. And we are able to see that when we spike the soils with the target compound serine, we're able to see a significant increase in the release rate from these coatings. So this is sort of where we're at with this project. We have the aptamers that recognize the targets, and we have the prototype that in the greenhouse is looking really promising. So now we're moving on to try to set up for field studies. We recently were able to get regulatory permission from the Canadian government. So Canadian Food Inspection Agency has a fertilizer safety unit. They've finally given us permission to set up a field trial, so we'll be doing that next year. And also we're interested in starting work at a smart farm that's just down the road from us here at Carleton. So this smart farm is basically a wired up farm, and we're interested in now using not just the smart fertilizer capsules themselves, but also the idea of the aptamer interfacing with a sensor that's permanently present in the ground and transmitting and working with the rest of the infrastructure that's present at the smart farm. Okay. So that story, I want to leave time for questions, so I'm not going to go too deep into the other stories, but that story is the one that we've been working on for the longest, how to integrate aptamers, with a smart fertilizer type platform. But more recently, and of course, thanks to the Grants4Ag program, we've been able to start exploring other areas where aptamers could play a role in improving agricultural efficiency, sustainability. One of them is using aptamers as a way to deliver something through a plant surface, for example, a leaf surface. So say a microcapsule like the one we discussed, or a nanoparticle, which we didn't discuss, that's coated with an aptamer that interacts with a surface receptor or some sort of surface feature that allows selective uptake of whatever is loaded in that capsule into that crop. So I won't talk too much about it because this is what we're actually using our Grants4Ag funding to explore. Still really new. So check this space for the future. I know that we're going to have some exciting things to talk about soon. And then the other area that we're interested in using aptamers in agricultural space is this idea that aptamers could be the treatment to the issue of, for example, stress. So heat stress, salinity stress. What about if the aptamer is more than just detecting these events, but is the treatment for these events? So we're working on aptamers that are basically mimics of, say, heat shock proteins. So these heat shock proteins have many roles, but among them is the idea that they could sort of bind to unfolded protein that's unfolded because of some stress response, and sort of protect those proteins and allow them to refold when the stress response has passed. So the idea that I'm just going to show you one small little bit of data, because we've got some of it protected, so it's safe to share. But we've developed for other scenarios aptamers that block protein aggregation or protect proteins from aggregation, and we would love now to apply this to agriculture. So what I'm showing you here, this is in the health space. We have aptamers that are able to block the aggregation of a protein that's implicated in Parkinson's disease. So what you're looking at the left is an electron micrograph of this protein, alpha-synuclein, that's been left to aggregate unchecked. And this is, related to the pathology of Parkinson's disease, and the death of dopaminergic neurons is related in some way. It's not 100% clear, but it's related in some way to this aggregation event. So you can see these tangles of fibrils that are present when the protein is able to aggregate without any inhibition. So no aptamer was present in this control. But on the right-hand side, this is another electron micrograph where we have the same protein, substoichiometric levels of aptamer that was designed or selected to block the aggregation event. And you can see we can shut off aggregation completely. So we have experience with this in the health space, and now we're looking to move that experience into the agriculture space and kind of combine all of our technologies together to really make an aptamer-enabled, smart farm in the future. So that's my story. I hope I can have time for lots of questions now. But the idea is, I hope that I've been able to show that aptamers really have a lot of potential. They are robust affinity reagents, and we can use them for all sorts of different applications. And they're pretty accessible. So the fact that they're synthetic, the fact that they are stable, this is useful for all sorts of different applications. I've shown you a little bit about some promising exudate aptamers that we've selected and some early prototypes of our smart release smart fertilizer type materials. But I think there's lots of room to innovate here. So totally interested to hear other people's ideas and other ways that we could explore or expand what we're doing into different space in agriculture. So with that, I have so many people to thank. Of course, the research is done by amazing graduate students, postdocs, technicians. Both this is from my lab, and over here on the right was Carlos Monreal's lab at the time of the start of our collaboration. I also have other collaborators, like Juan Schneider was involved in helping us with the coding. So he's the CEO of a small company called Nano Grande that's in Montreal, Quebec. And he's able to help us to kind of integrate our aptamers into a biocompatible coating that we were able to use for our prototypes. And of course, I have to, again, just one more time, thank Bayer Crop Science for the opportunity to speak to all of you, and of course for the Grants4Ag Award, and you for your kind attention. And I'm really, really excited to hear your thoughts and to answer any questions.
Oh, thank you, Maria. That was great. I really appreciate it. There's a few, there's 2 or 3 questions in the chat, but we only have a small group here, so I would invite you guys to unmute and turn on your cameras if you like, and we'll do the questions that way. Simon, I can see you, so why don't you jump in and go first?
I'll go first, but I'll make sure I hand it back to Tien Dung Lee afterwards, because he was the first question in the chat. But I'm happy to kick this off. And Maria, on the mycotoxin example, you showed 2 false positives. I'm interested in how many false negatives were in the test.
Great question. So in that small blinded field trial, we didn't have any. We've now expanded. Obviously, this was now a few years ago, and now we're in the middle of scaling up. So now we're in the realm of hundreds of tests that we're able to print at once and able to use it. And we're still seeing very low levels. So I think in the last trial where we were doing 50 samples, I think we had 1 that was false negative and 1 false positive. What we're seeing sometimes, so the false positives, these were really, really contaminated samples. So, you could look at the sample and say, oh my gosh, this definitely has, mold. And I think maybe that kind of confounded the nanoparticle part of the test. So I think that was going on. And then I think the false negative is still, it's going to be kind of what you would imagine. It's a low level. Our aptamer, maybe there, because of the matrix, is not able to recognize the presence of that toxin. So we're still working on, keeping them as low, getting them as high affinity as we can. But still, it's looking quite promising. So we'll see. This next, we have funding for the next 3 years to basically push this out now to the grain elevators. So the idea that we've been doing this all in our lab, which is fine, but now, like, what if it's in someone else's hands? Do the false positives and negatives go up and all that sort of thing?
And I'm sure you've heard this comment before, but in
Yes.
this example, it's the false negatives that concern me. So keeping a low level of false negatives is the right side you want to be on. The false
Absolutely.
positives wouldn't concern me so much.
I agreed. So I guess it's more like an economic issue if you say a lot is not, let's not go ahead with this lot because it's too high, and then it's not. Okay, so it was a waste. But a false negative, right? If you let a lot through, that does end up, then there's reputational issues, there's cost, there's health. So you're 100% right. You're 100% right.
Yeah. And just to give you some insight, sorry, Phil.
No, please. So great. Yeah.
Just to give you some insight from Europe is when you're doing these sorts of tests and whatever you do, you have to send it off to a lab for a validation, a GLP
Exactly.
lab for validation. And I don't mind sending off a false positive and getting a negative reply and still being able to sell the corn. So just reinforcing everything you said. Thank you, Maria. And also, I forgot to thank you for a wonderful talk. I really enjoyed that. Sincerely, I really enjoyed that. Thank you.
Thank you, Simon. That's great. So I do see the other questions. I could just maybe jump in. On the detection limits at the mycotoxin example, do you also refer to current allowable limits or just try to get as sensitive as possible? Okay, great question. So I think you mean when we have a mycotoxin, we're going to start a selection. Do we just try to get as sensitive as possible, or do we just aim for whatever the limit is? So obviously, for most cases, we're trying to get as sensitive as possible. So we're trying to make the selection as stringent as possible. And so that way, even, obviously for the application, but also when you're trying to eventually publish something, you want to say, oh, it's the lowest limit of detection that is out there, blah, 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 right? So obviously, but practically, when we're choosing a system to work with, we're looking at what the allowable limits are. We're looking at what's achievable, because you obviously we're limited in what we can do, so we want to tackle problems that we think we have the capability of solving. So certainly it comes into play when we're making a decision. Not every case has to do with the most sensitive aptamer possible, and that, like, for example, the protein aggregation case was one where we weren't looking for the aptamer that was able to bind the lowest level of the protein. It was the aptamer that was able to block the aggregation. That was what we were looking for. So actually that happens at higher levels of protein. So we didn't want to block or interact with the protein at low levels. So depending on what the scenario is, when we're designing our experiment, we may be going for the most sensitive thing possible, but we may not be. So it's case by case.
Yeah, thanks, Maria. Yeah, that's my question. Yeah, because sometimes we in publication we may want to go for as sensitive as possible, like maybe breaking the boundary, but then
exactly.
In application in the real world, the usability of the test is really about what the regulation is, and then this is past that regulatory level, or it's just like not right. Sometimes we don't have to be like to get headache and go to that boundary. It's already work. So that was my comment. Thanks.
Such a good comment. Absolutely. It's always fun to be like, yay, Mount Everest, like we did it because it was the best. But then you do have there's a certain point where it's limiting. You're not getting the gain that you, for all the effort that you need to make it more sensitive. It's maybe not worth it. So good point. Okay, let me just go see. Oh, great. So Ingrid asks, what are the benefits of DNA aptamers versus RNA aptamers? Great question. So actually, I think for a long time, there was a perception that RNA aptamers, like RNA as a biomolecule, is more built for this kind of thing, folding up into a structure, interacting with the target, that kind of thing. Whereas DNA is supposed to be double-stranded, one structure, not, malleable, blah, blah. So for a long time, there were a lot more RNA aptamers than DNA aptamers. We have a database so that of all aptamer selections, from 1990, I think it's till 2015, and then we're continuing to update. And we looked at, just statistically, was there any difference in the average RNA aptamer affinity versus DNA aptamer affinity? We didn't find any statistical difference. So I think DNA aptamers and RNA aptamers, they can both bind their targets, high affinity, all that sort of thing. I think it's more application. So if you're doing something, there's a lot of interest in things like synthetic biology, in vivo, in cellulo type assays or applications, then I would say RNA makes more sense, right? Because then it's going to be a lot more straightforward to have them present, in that environment. If you're looking at something that's going to be on a shelf for a long time, or in soil, or in a complex matrix, then maybe it makes more sense to use DNA. It's a little bit more stable. But even so, you can make chemical modifications to RNA aptamers to make them more stable too. So I think it's depends on your application.
Hey, well,
go ahead, Ingrid.
Yeah, it was a really interesting talk, and I just love people working more on DNA, because I think DNA is so much more than just storing genetic information. So this is I'm really excited about the work, and I also agree. I was asking just because it seems like straightforward to use DNA. It's more stable. It's more. It's cheaper. I think, and I think it's also not degraded. Like, it's stable on many levels.
I agree.
Inside the cell and outside the cell and whatever. So I think this is really exciting. Maybe I can ask one more question if no one else is asking. So what about the structural variety? I'm a structural biologist, and you showed a couple of nice little pictures. Do you know the structures of the aptamers that you're working with? How straightforward is it to solve the structures? How big is the variety?
Fantastic questions. So in some cases, we have a lot of circumstantial evidence about the structure of the aptamer, right? So we can do things like circular dichroism, have some idea of global structure. We can do kind of footprinting experiments, understand what is bound up, what's not, what's available, what changes when the target binds. Structural experiments like NMR experiments, X-ray crystallography have been done on aptamers. They're just challenging. Growing a crystal is challenging. NMR, you do need a lot of sample, and if the aptamer is a little long, you probably might have to label. So we don't necessarily do that. We have tried to collaborate with others who are more expert in that. But for the most part, we have, like I said, the circumstantial kind of evidence to try to understand how the aptamer folds, and then use that to design whatever it is that we're trying to do. The structure, I think this is something that is overlooked a lot. So in many cases, an aptamer, is published, it does something, and there's maybe no idea of how it actually folds. And then that just makes it harder to translate, because if you don't know how it folds, it's hard to, turn it into a sensor. It's hard to understand how the environment will change its structure, all that sort of thing.
Thanks. So I will send you an email, because I think we published a paper on something where we developed a method where we think we can crystallize nucleic acids.
Perfect.
But we don't really have too many examples to try, and our examples failed because it's really large. So I would be really interested if we can use this actually maybe with an aptamer. So I think maybe we can just chat at some time and see, because I just have a feeling that it could be really great to try, and would not be that much work on our end to just give it a try, and then maybe
let's do it. Yeah, this is a big challenge for us. So we don't have that expertise. So if you have an approach, we would jump on that, absolutely.
I'm going to send you the paper, and then maybe we can set up a meeting and discuss that.
Perfect. Sounds great.
Outstanding. Any other questions for Marie, or anybody else like to jump in?
Hello. Yes, I have a question. Andrzej Kurenda, I'm a biophysicist. So I have 3 questions, but I hope that they are easy to address. So first of all, I'm really interested if you know something about the limitation of your technology. Are there any groups of molecules which you cannot target? So this is my first question. Second question is, how long development of an aptamer for specific molecule takes? It's a more practical question. And third question is, I didn't get from your presentation how you really measure affinity between the aptamer and the molecule.
Great question.
Thank you very much.
Okay, so all great questions. I'll start at the end. So how do we measure affinity? And then we'll work backwards because I probably already forgot the first question. How do we measure affinity? Again, it depends on the situation. We try to measure by a number of different approaches because often what ends up happening is that not every technique works for every aptamer. And this was one of the, in 2015, we published a paper in Analytical Chemistry where we looked at, a family of aptamers for one specific target. They all had, papers, some were our own, that showed that the aptamer could bind the target. But then when we did a suite of techniques, inevitably certain techniques would come back with a negative, like this doesn't bind under this scenario. So we try to do many different techniques. And what we try to keep in mind is, different scenarios. So the aptamer is tethered and the target is free. The target is tethered and the aptamer is free. Both are free in solution, and try to find techniques that mimic all these scenarios to better understand what we can use as an application, right? So if the aptamer is tethered and the target's free, that might be something like, surface plasmon resonance. We put the aptamer on the surface, we flow the target through, and we look for changes. Both things free in solution. I showed you microscale thermophoresis. This is a technique that basically measures movement in a heat gradient that's affected whether there's a complex or not, essentially. So that's both things free in solution. And then say the target is bound, we might put the target on a magnetic bead and a labeled aptamer, and then just basically do an experiment to see how much aptamer is retained by the when you use the magnetic bead. So there are a few different ways that's done, and case by case depending on what the application is. Okay, so that was one. How long does it take to generate an aptamer? So I think a safe bet is that it takes a few months. We've had aptamers that, within 3 rounds, which around, if nothing else is going, a few days to a week to do a round. But, I have all students, we're in a university setting, so they have TA and everything. So sometimes things take longer, but it takes a few days to do a round. We've had some scenarios where 4 rounds and we had like such great enrichment, we couldn't believe it, and we actually found sequence. We had a more challenging, like the serine example. It took months and months and months. We had so many false starts. And I think this relates back to your first question: is, are there limitations? Yes, there are limitations. Size of the target plays a role, and I think that's just number of potential molecular interactions that the aptamer can form with the target. So proteins, for example, tend to have better binding affinities than small molecules for these aptamer systems. When you're looking at the target that you're thinking of developing an aptamer for, imagine that it's a nucleic acid that's going to interact with it. If there's no hydrogen bonding possibility, if there's no pi stacking possibility, if there's no electrostatic, like a siloxane or something like this, you can imagine it would be challenging to find an aptamer that would interact with it. So then you have to think more creatively. There are things called basically somomers. So they're short off rate modified aptamers. So aptamers that have been modified with side chains that have hydrophobic type groups, that you could use something like that to make a hydrophobic pocket for something that's less likely to interact with something like just a regular DNA strand. So that's a challenge for sure. If you're looking at the target and it doesn't have those juicy functional groups, then you think, oh, maybe this is going to be a really bad idea to start an aptamer selection without being creative.
Great, thank you very much.
You're welcome.
All right, last call for questions. We're coming up on the top of the hour. It's been a great. I'm kind of amazed the time for questions has just flown by. There's a great discussion there. So Maria, thank you so much. That really was wonderful, and I'm so glad the group here enjoyed the presentation and we had such a great discussion. So if you have enjoyed it, I would encourage you to follow along on our LinkedIn page. You'll see the details up on the slide here. Just jump in, join the conversation, and stay aware of programs like this that we'll be offering through the year. And we hope, as I said, we hope to do this about once a month. And now that it's been a roaring success, the first one, I have no doubt we'll be able to do so. So with that, thank you, Maria. Thank you all for joining, and very best wishes for a good day.
Thanks, everyone. That was great.
Thank you. Thank everyone. Bye-bye.
Bye.
Thank you.